According to the American Cancer Society, the estimated total amount of deaths due to cancer have significantly risen over the past 40 years. Specifically in 2014, 36,000 people were diagnosed with liver cancer, and over 24,000 people died from liver cancer-related causes. Cancer is the result of growth and spread of unhealthy cells within the body. To combat such a viral disease, alternative methods must be utilized as eclectic ways to track the spread of liver cancer at an early stage for preemptive methods. Computer modeling, specifically computer vision and medical imaging, has shown to be viable methods to track the spread of various cancers. Pathology imaging is aimed towards developing computer software to track the probable growth of cancers by using various algorithms. Such applications of biotechnology can be an alternative for combating viral diseases. In this figure, we see the intended image analysis process for detecting abnormalities within the tissue by looking at the nuclei. Using image analysis techniques can develop signatures to represent patient-specific tumor morphologies through the analysis of millions of cells and digitized whole slide images. Clustering of these signatures aggregates tumors into groups with cohesive morphological characteristics. For instance, in the first step, researchers would first scan slices of tissues to create high-quality and resolute images of the slide that can clearly show the nuclei and its positioning. Next, in a semi-automated process, the nuclei are differentiated from the tissue in order to be used for detection of abnormalities within the tissue. This process is done by looking at whether or not nuclei have irregular parameters such as diameter, circularity, and perimeter. In the final and most important step, a thorough analysis of the data is critical to the success of pathology imaging. In this case, we would look at comparisons with other databases to see the significance of the results, and whether or not they are consistent or irregular from other results. This collaborative effort is a critical piece in pathology imaging, as the results of others can often tell the outcome of a certain patient. This is just one scenario of pathology imaging, as there are various algorithms that can be utilized for medical imaging. One such algorithm is image registration. Image registration is an image processing technique used to align multiple scenes into a single integrated image. As you can see in the figure, it helps overcome issues such as image rotation, scale, and skew that are common when overlaying images. Image registration is often used in medical and satellite imagery to align images from different camera sources. Digital cameras use image registration to align and connect adjacent images into a single panoramic image. Registration is necessary in order to be able to compare or integrate the data obtained from these different measurements, which is often needed in neuroimaging scenarios. In medical image registration, many studies are applicable, including change detection, tumor monitoring, and deformation growth. This can only work if there is an extensive dataset for a patient. For instance, here the source image is registered with the target image to create the registered image of the brain. When using further analysis such as a saliency map, we find the growth of abnormal tissue, because we are able to combine these images to find the differences within the images. Often in neuroimaging, the use of image registration involves elastic or non-rigid registration to cope with deformation of the subject because of cases of breathing, anatomical changes, and maturation. Different from rigid registration, to extract the cross-section, the algorithm creates a histogram to find changes in vector positions to try and create a consistent tissue image. Similar to the SIFT algorithm, this histogram accounts for rotation and variance within the image. As you can see in both the rigid and non-rigid registration, you still get the same pathological image. Here is just a short video detailing both the rigid and non-rigid registration, and the differences between the two. In 2011, Kong tested a complete and novel workflow for quantitative nuclear feature analysis of glioblastoma using a high-throughput whole-slide microscopy image processing as presented as it relates to treatment process and patient survival. With a complete suite of computer algorithms, large numbers of microanatomical structures, in this case nuclei, are analyzed and represented efficiently from whole slide digitized images with numerical features. The computer analysis presented a better discrimination than traditional neuropathologic review. As you can see by this graph, the use of computerized methods significantly discriminated between clusters of nuclei in glioblastoma. As a result, his analysis method showed a better understanding of disease progression for glioblastoma. Charles Hayes utilized non-image registration techniques for breast cancer detection and MRI images. He presented a new approach for the non-image registration of contrast-enhanced breast MRI. He compared non-rigid registration with affine and rigid registration in finding deformities in breast images. He finds that rigid and affine registrations are very inefficient in finding deformities in cancer imaging. His results clearly indicate that non-rigid registration algorithm is much better to able to recover the motion and deformation of breast rather than rigid or affine registration algorithms. This is because non-rigid transformation allows more flexibility when finding irregularities, as shown by these two figures. Finally, Nakamura in 2014 tested the registration between pathological image and MRI image for comparing different modality images of brain tumors. Magnetic resonance imaging is the preferred modality for brain tumor detection and preoperative localization. However, invasive regions of tumor are often unclear in MR images, and thus it is difficult to find tumors from MR images. Nakamura compared rigid and non-rigid registration to see which algorithm could effectively resemble an MRI image. Similar to Hayes, he found non-rigid registration to work better due to its flexibility. This summer I'll be working at Stony Brook University under Fusheng Wang at the Biomedical Informatics Department. Several techniques and instruments are required for the study that are readily available in Dr. Fusheng Wang's lab. First, although a digitized tissue scanner may or may not be readily available at Dr. Wang's lab, most people in this lab use OpenPace, which is an extensive pathology portal created by a collaborative effort of many doctors around the world. It contains a vast and diverse array of tissue images from many patients that is more than suitable for this project. Next, understanding and use of MATLAB language, which is a technical computer language, is required to fully utilize image registration techniques for this project and for further image analysis. Luckily, Dr. Wang has access to MATLAB software and will be teaching how to use MATLAB software for image registration purposes. Thanks for watching.